Then we use an example from the mouse, like a human chimpanzee comparison, and that's the example I mentioned a few minutes before, is the one we use the mouse as the algorithm. Okay, so that idea is like this. So this is like the hypothesis 1 and hypothesis 2. So we have a null hypothesis and the alternative hypothesis. Then the null hypothesis here is showing there's an equal rate between human and chimpanzee lineages. Then the alternative means they are different. So that's one of the lineage will show much higher amino acid substitutions indicated by DN. So that's the DN large in DS, and using then compared to the other one. So the hypothesis listing here show you exactly we are testing. We are testing whether the evolution rate on the human lineage is faster than our counterparts, the chimp lineage. Remember, just a few slides ago, we talked about the Ka-Ks ratio between human and chimpanzee, and we already identified, people already identified genes show higher Ka-Ks ratio with that comparison. Now, with the branch comparison, we're trying to ask whether the fast evolution is occur only on one of the branches, or at least one of the branches is much faster than the other, given the same evolutionary time, because they evolved from the same common ancestor, then derived to two different lineages, give rise to two organisms present today. So that's the model we are testing. And this is the table from the paper. So now you can see the estimates has two estimates. They compare the chimp lineage compared to the human lineage. And with this comparison, there are two separate parts in the table. We could identify certain genes evolve faster in the human lineage, and maybe the other set of genes evolve faster in the chimpanzee lineage, and using the mouse as an outgroup. So this is the idea, to use the test model test, and to show the evolution rate differences between the human and chimpanzee lineage. And so if you look at the function of these genes, so this is the functional category. So that's another way to analyze the data. So previously we could look at individual gene category. So we say, okay, A gene shows whatever number estimates and whether it's significant or not. But here we pull the genes together into functional category, and we want to ask whether there's a certain type of genes, as what we mentioned previously, like a male reproductive protein, are evolving at much higher rate than the average of the genome, or than the rest of the genome. So that's another way to analyze the data, pull the genes into different functional categories. And the common practice now is to use the gene ontology, or like the functional group of the genes, and there are certain ways to characterize that. Then you can pull the genes together. The second column is the number of genes in each category. Of course, you can show there are some category have a large number of genes, some category only have few number of genes, but overall, you can use this average among genes in the same category to compare, to do the same comparison between the two close related lineages. Well, you probably would already know mouse is really far from human and chimpanzee. So the better estimate will be from the close related outgroup, and that's leads to the sequence, the genome sequence of the macaque. And I'm not going to talk about the detail of that paper, but that has been like a reanalyzed. So with the macaque sequence, all the human chimpanzee comparison have been reanalyzed, in the overall signature is pretty similar, but with more detailed analysis. So that's like using a closed outgroup is always better than a distant related outgroup. And in your study, that's the same thing. If you want to test the two lineages, you want to have a sister group closely related to the two taxa you are interested. So that's just sort of like a common practice. But previous paper at that time, when they compare the human and chimpanzee data, there's no macaque sequence genome available. That's why they use the mouse as an outgroup. Then there's a lot of paper talk about the positive selection or the signature of positive selection in the human evolution. And you can look up, I only put a few here, but in general, people are interested in what the differences between the human lineage and our close related chimpanzee lineages. And the signal is very similar. So showing here is the summary of from another group which did very similar work. And they also, so this is just for you to understand or just to experience how people did the analysis, and different people using very similar data set with slightly different method, or use very similar method and but recalculate or reanalyze a lot of things. So here is the data they used. Use the close relative species in the outgroup and look at the chimpanzee human differences. Then they also compare those genes show positive signature of positive selection, whether it related to human disease. So that's a very interesting category. So that means we have some mutations, genetic mutations, and those mutations affect human health. So they are disease mutations segregating in human population. But in those cases, or in some of the cases, we detect positive selection. So although the p-value now is like a 0.49, but that's the pool data with all the known disease allele or disease genes. So that's a very interesting topics in molecular evolution. Why some of the disease alleles are still segregating in the human population, and some of them, although they are not very deleterious, but they are evolving at a certain frequency in human population. And historically, a proportion of these genes might be under positive selection. So that means those alleles used to be advantageous during the human history. And there are some specific cases we could discuss for each category, but you could also look up the paper and other discussions on this subject.